Welcome to the Explainer. Right now, literally as we speak, inside your body, millions of your cells are actively deciding to die. But how exactly does a microscopic blob of biology, something completely devoid of a brain, make that ultimate high-stakes choice between life and death? It's a genuinely fascinating, incredibly intricate decision process, and today we're going to break the whole thing down.
So, think about it for a second. When does a cell finally just throw in the towel and say, okay, it's better to die than to survive? Now, you might assume that the second a cell gets damaged, it just pops or maybe shuts down like a broken machine. But actually, that couldn't be farther from the truth. In reality, cells possess an amazingly tenacious instinct to survive massive amounts of stress. They really don't want to die. So when does a cell finally conclude that pulling the plug is the only option left?
Let's dive right into Section 1, the tenacious instinct for survival. See, before a cell even gets close to dying, it mounts a fierce, multi-layered defense. The common myth out there is that a single stressor acts like an instant kill switch. Boom, dead. But the reality? It's way more resilient than that. If a cell suddenly lacks oxygen, it doesn't just die right away. It shifts its entire metabolism. If its proteins misfold, it actively tries to refold or degrade them. If its DNA gets damaged, it literally hits pause on its life cycles to attempt repairs. Basically, between cellular stress and cellular death, there is this massive buffer zone filled entirely with adaptation and repair systems.
And to manage this buffer zone, cells are constantly monitoring a sort of internal checklist. They're checking their DNA integrity, their mitochondrial health, their overall energy or ATP status, and their redox balance, which is essentially a measure of how well they're neutralizing toxic oxygen byproducts. They're also keeping a close eye on their ability to manage proteins, plus listening for external survival signals from their neighbors. And a really crucial thing to remember here, this isn't some conscious thought process. We're talking about a highly complex biochemical network that's constantly shifting and calculating in real time.
Moving on to Section 2, intensity and duration of stress.
It turns out it's not just about what kind of stress a cell is facing, but for how long it lasts. Actually, a brief initial stressor can be a really good thing. For example, a slight temporary bump in reactive oxygen species, or ROS, acts as a helpful signal. It tells the cell to adapt and build up better antioxidant defenses. But if that exact same stress is prolonged, well, then it totally overwhelms those defenses. The damage just keeps accumulating until it hits a critical point, transforming what was a manageable burden into a lethal one.
So, a cell's decision pathway isn't a simple one-way street. It's more like a multi-step crossroads. First, it senses the damage. Then, it integrates that data across its entire internal network. Step 3 is the real make or break moment, the recovery check. Is recovery still possible? The cell might choose to pause and repair. It might adapt, or it might even enter a long-term dormant state called senescence. It's only if all those recovery options completely fail that it finally takes the exit toward death.
Which brings us to Section 3, the threshold of life and death.
Okay, this is absolutely the climax of our explainer. The exact moment a cell transitions from desperately trying to survive to actively executing its own demise. We call this the point of no return, or simply the threshold. But here's the wild part. This threshold isn't some fixed universal number for every cell. It's a constantly moving boundary. The threshold for a highly resilient cell in your intestines is completely different from, say, a super delicate neuron in your brain. It depends entirely on the cell's initial state, its current energy levels, and its specific job in the body.
To put that into perspective, picture a microscopic set of scales. Survival signals are piling up on one side. Things like growth factors, ATP energy, active DNA repair mechanisms, and cellular recycling, known as autophagy. But then, death signals are weighing down the opposite side. Severe DNA damage, calcium overload, massive oxidative stress, or a huge traffic jam of misfolded proteins called ER stress. As long as that survival side stays heavier, the cell lives. But the absolute second that cumulative damage outweighs the protection, that threshold is permanently crossed.
And here's a crazy fact. A cell doesn't always need some violent toxic attack to die. Sometimes simply losing its connection is enough. If a cell detaches from its proper tissue structure and stops hearing those "hey, stay alive" signals from its neighbors, it triggers a very specific type of death called anoikis. It's actually a brilliant evolutionary failsafe. It prevents rogue cells from floating away and growing out of control somewhere else, which, by the way, is exactly what cancer tries to do.
Let's shift gears to Section 4, role of mitochondria and energy. So, you might be wondering, who exactly is doing the accounting for all these life and death signals? Let's take a look at the cell's central command center.
We've all heard mitochondria called the powerhouse of the cell, right? But they are so much more than just a mindless energy factory. They're actually the central sensors reading the overall health of the entire cell. They regulate calcium, they generate those reactive oxygen species, and they actually physically hold the signals for apoptosis or programmed cell death. Basically, all the major stress pathways intersect right there at the mitochondria.
And this is fascinating. Energy levels literally dictate the style of cell death. Because, as it turns out, dying gracefully is expensive. If a cell has plenty of ATP energy left, it can execute a highly regulated, precise demolition, neatly packaging up all its contents. But if it goes totally bankrupt, like rapidly losing all its ATP during a severe injury such as a stroke, it can't afford to pay for its own demolition. So it swells up and undergoes this catastrophic, messy collapse that just leaks out and damages all the surrounding tissues. Because this entire process is so ridiculously complex, people often fall into the trap of looking for a single Grim Reaper molecule. You might hear about famous proteins in biology like p53 or BCL2. But listen, no single molecule makes this decision all by itself. I mean, the protein p53 can trigger life-saving repair in one context and then trigger death in another. The ultimate fate is always decided by an entire, highly interconnected biochemical network tipping into a brand new state.
Finally, let's wrap up with Section 5, the boundary-changing disease.
If we zoom out from the microscope for a minute, how do these microscopic cell fate decisions actually apply to the human body? Well, it turns out that our most devastating diseases are essentially errors in this exact decision process. Cancer happens when cells abnormally raise their death threshold, surviving massive DNA damage that absolutely should have killed them. Neurodegenerative diseases? That's when neurons slowly lose their adaptation capacity over a lifetime, eventually crossing that death threshold. And ischemic diseases, like a heart attack, involve a rapid drop in ATP, triggering that incredibly messy catastrophic cellular death we were just talking about.
Understanding all of this really highlights why the timing of medical intervention is absolutely crucial. In the very early stages of a stroke, the brain cells are heavily stressed, for sure, but they are still in a reversible state. If doctors intervene right then, they can save the tissue. But once that boundary, that critical point of no return, is crossed, the cell is locked into a self-amplifying, irreversible death spiral. At that point, applying the exact same treatment is going to do virtually nothing.
Which leads us perfectly to the core philosophy of our explainer today. Cells do not die simply because they are damaged, but because the damage exceeds their capacity to cope. Cellular death isn't just some random accident of nature. It is a highly calculated, final biological choice made at the very end of a long, desperate struggle to survive. So the huge takeaway for the future of medicine is this. Treatment isn't just about throwing drugs at a problem to kill bad cells or save good ones. It's actually about shifting these invisible cell fate boundaries in our favor. It's about figuring out how to lower the threshold for a cancer cell so it destroys itself, or artificially raising the threshold for our neurons so they can survive into old age. If manipulating this very threshold is the frontier of medical science, we really have to ask ourselves, which boundary are we going to learn to move next? Thanks so much for joining me on this explainer, and remember, keep questioning the science around you.